Hello. Today I'll be discussing my work with characterizing the function of the amyloid precursor protein in an Alzheimer's normal cell model. To begin, we must define what Alzheimer's disease is. Alzheimer's is a mental deterioration of the brain due to generalized degeneration. This means that healthy neurons are destroyed and unable to communicate between regions properly. This is defined by amyloid plaques developing in the brain, along with neurofibrillary tangles, destroying synaptic connections. Although the cause of Alzheimer's is unknown, one large theory exists, the amyloid beta theory. This theory proposes that a lack of amyloid precursor protein in degenerated brains causes amyloid fragments to produce in the brain. These plaque aggregations clog the brain's pathways and destroy connections. However, the amyloid precursor protein's function is largely misunderstood. The role of the neuron is actually unknown. An APP in the neuronal membrane is directly related to the amount of membrane-embedded cholesterol. The C-terminal transmembrane domain of APP actually helps cholesterol bind to the membrane. With this new information, we can assume that the amyloid precursor protein actually has more of a direct involvement with cholesterol rather than without. This difference might be the reason why the amyloid beta theory is not the actual culprit of Alzheimer's disease. How an imbalance of cholesterol may affect the brain function is through two main regions, the membrane and the astrocyte. The membrane is important to the cell because it allows for proper transmission of neurotransmitters. The cholesterol is very important for the fluidity and structure of the membrane itself. If the membrane's shape is compromised, then neurotransmitters are released in the wrong places and bind to the wrong receptors of neighboring neurons. If this happens, then the cell cannot communicate properly and behavior is altered. The astrocyte is another important feature of the neuron. The astrocyte is where the cell is able to sit and rest in the proper sequence along the path of connection. The astrocyte uses a cholesterol equilibrium to maintain the placement with its associated neuron. If cholesterol is not binded to the membrane properly, excess cholesterol may congregate within the body of the cell. When this happens, the equilibrium between the astrocyte is no longer maintained, and the cell may shift out of place along the astrocyte. This shiftment might destroy crucial connections that could possibly code for the proper memory, cognitive, and physical function of the Alzheimer's disease patient. Our research question asks how the removal of the amyloid precursor protein could affect cholesterol development in human neurons. Effectively, we are simulating an environment where the amyloid precursor protein has degenerated and no longer functions properly. Our first aspect of methodology is actually creating these neurons. Using iPSCs, or induced pluripotent stem cells, we can differentiate our cells without the presence of the APP gene. When this occurs, we're able to simulate an environment where a neuron has degenerated into improperly producing the amyloid precursor protein. First, we used artificial signals to test whether the potassium and sodium currents were healthy or not. Once we found these voltage units were aligned with potassium, we knew we had created a simulant cell. Externally, we used CRISPR to remove the APP gene. Although this work was not part of my actual involvement in the lab, it was crucial in creating our two different groups, the APP knockout neurons and the wild type neurons. Once we had differentiated our neurons, we were able to stain each neuron using a Philippine dye and an AM143 dye. When we stain these neurons, we were staining both cholesterol and the membrane in order to differentiate between what cholesterol was binding to the membrane and what cholesterol was not. This staining allowed us to quantify the actual amount of cholesterol remaining in the neurons. Next, we had to image the cells in order to establish where the cholesterol had been produced and where it had been unbinded. We used an Olympus IX48 camera to capture the neurons at 3 different stages: 30 days in vitro, 60 days in vitro, and 90 days in vitro. These 3 points are the 3 most crucial points in neuron development. At 30 days, the neuron is early, it's maturing still, but not fully developed. At 60 days, that is a crucial date in which the neuron is fully functioning. 90 days allows us to see the direction the neuron is growing in. We used image data analysis to establish where the cholesterol had grown. We overlaid the RGB values of cholesterol compared to the membrane in order to differentiate between what white marks were cholesterol and what white marks were membrane. Our first set of results compare surface and total cholesterol at 30 days in vitro. At 30 days, we saw that the surface cholesterol had grown more than the wild type, had been expressed more in the wild type as compared to the knockout. We also saw that in total cholesterol, the knockout neurons had expressed more cholesterol than the wild type. On these images, we see the green represents cholesterol, and the blue represents the membrane without cholesterol. At 60 days, on the surface, the wild type expressed more cholesterol than the knockout group. With regards to total cholesterol, the knockout produced slightly more cholesterol than the wild type. It is important to note these 4 asterisks representing a p-value of less than 0.05. At 90 days, the surface cholesterol in the wild type group was higher in value than that in knockout. At 90 days, in total cholesterol, we saw the wild type group express more cholesterol than the knockout. This y-axis represents the intensity of color overlay expressed. This intensity is quantified in order to express a level of cholesterol compared between both the wild type and knockout groups. 2 asterisks represents a p-value of less than 0.0005. As we compared our results, we saw 2 main takeaways. The knockout had significantly more total cholesterol than the wild type on day 60, but the wild type had significantly more membrane cholesterol than the knockout on 60 days. It is important to note 60 days once again because this is the first day that the membrane is properly executing its functions and connecting between other membranes, between other neurons. So what we can conclude is that the APP knockout was unable to bind cholesterol to the membrane. This would explain why there was more cholesterol on the surface in the wild type group and more total cholesterol in the knockout group on day 60, because the unbinding cholesterol congregated within the body of the cell. We need to recall the role of the APP that we have proposed, that it binds cholesterol to the cell membrane. When this is unable to happen, when the APP is degenerate and functioning improperly, the membrane of the neuron is compromised and the function is therefore unable to execute it properly. The neurotransmitters are fired at an imbalanced rate, and the cell will shift off its astrocyte when there's too much total cholesterol. The fact that there was a difference in the amount of cholesterol between the knockout and the wild type groups highlights the function of APP as binding cholesterol to the membrane. In the future, we need to increase the number of cells as well as our functionality of the APP. Thank you for listening.